Good morning, everyone. I guess we get started. Thank you very much for joining us this morning and this early in the morning for our breakfast seminar. I am Fabia. I'm a business development manager at Biognosys, and I'm excited to be the chair of this exciting seminar on how to enhance your plasma research with Biognosys PQ500 Reference Peptides, featuring two very exciting speakers. First, I'll give you an overview on Biognosys and the products portfolio, and then an introduction on PQ500 Reference Peptides Kit. I will then leave the stage to my colleague, Sebastian Müller, principal scientist at Biognosys and developer of the PQ500 Reference Peptide Kits. He will talk about the use of PQ500 in large-scale studies using SureQuant. We will then leave the stage to our guest speaker, Manuel Meyer, a professor in cardiovascular proteomics at King's College London, who uses PQ500 to identify biomarkers for mortality prediction in COVID-19 patients. Biognosys has been leading innovation in proteomics for over 13 years, thanks to a strong scientific foundation and R&D-centric mindset. We are inventor and developer of unique proprietary proteomic solutions, and to date, over 2,000 publications feature Biognosys technology. On top of that, we are one of the largest facilities in the world for large-scale discovery proteomics. This allows us to serve our ever-growing global customer base, thanks to our contract research services and product solutions. As part of our products portfolio, we do have our software solutions for different proteomics applications. Spectronaut is our flagship software for DIA. We then have SpectroDive for targeted proteomics, SpectroMine for DDA and isobaric labeling quantification. And last but not least, our free software for QC, Quick, that works together with the IRT kit. As part of the kits, we then also have the sample preparation kit for seamless and streamlined sample preparation and PQ500. PQ500 enables the absolute quantification of hundreds of human plasma and serum proteins across the entire dynamic range. It targets FDA-approved clinical markers and so allows comparable results across cohort studies, platforms, and laboratories. It is compatible with different acquisition methods and depletion protocols. The kit contains 804 purified and AAA quantified stable isotope label standard peptides, cis peptides, that were selected experimentally upon robust tryptic release and high stability. The kit also contains the IRT standard peptides and so allows assay transfer across different LC setups and also QC readouts with Biognosys software solutions. The kit, the cis peptides are either heavy arginine or heavy lysine, contain either heavy arginine or heavy lysine, and therefore can be distinguished from their unlabeled endogenous counterpart. And our software solutions, SpectroDive and Spectronaut, automatically calculate the ratio between the two channels and so allow to distinguish them, and also the software display the signals of these two channels next to each other. So yeah, PQ500 has been designed, is a big panel. It has been designed to be acquired also in data-independent acquisition, DIA mode, and data can be very easily analyzed with Spectronaut. And for targeted proteomics approaches, MRM, PRM, and SureQuant, we do have SpectroDive. And I think it's going to be very nice now that our two speakers will show both applications in their presentations. I also take the chance to let you know that right now we do have a time offer upon purchase of PQ500. And basically, you can access for free either 6 months of SpectroDive or 3 months of Spectronaut. So if you are considering to give it a go, it is a good moment. And if you have any questions on that, please visit us at booth number 2. You can also always go to our webpage, biognosys.com/PQ500. And at the very, very end of this seminar, we will have a Q&A session where you can ask anything. And with that, I gladly leave the stage to my colleague, Sebastian.
Thank you, Fabia, for the kind introduction, and thanks all of you for coming to our seminar this morning. Today, I want to share with you the results of a study that we did where we measured the same set of samples on different LC-MS instruments, and to demonstrate you the reproducibility and consistency of the data you will get. So what I will talk about today is I will quickly introduce plasma proteomics and PQ500, and then briefly talk about the methodology we are using in this study. Then the multi-site case study itself, where we use a small prostate cancer cohort, and then some outlook and conclusions. Quick intro into the human plasma proteome. So the human blood consists half and half of blood cells and the liquid part, which is called the plasma. Plasma actually contains mostly water, but also 7% proteins, small molecules. But since blood is flowing through all parts of the body, these proteins are very nice to investigate actually the health state of the body. And with even a small draw of plasma, you get out a lot of sample material, so you're really not limited in your sample amounts. There is a but, so the protein composition in plasma is very uneven, so it has a super high dynamic range with only very few proteins making up the biggest mass of the plasma proteome. So you really need to sequence through all these high abundant proteins before you get to the lower, medium, and low abundant regions. But since it's so available and attractive, nevertheless, the number of plasma proteomic studies, and this is already from 2016, steadily increases over the years. So people come up with more and more methods to tackle the plasma proteome. The number of plasma studies do increase, and also now several labs are working on the same phenotypic subjects, which makes a comparison of studies actually more attractive now over time. And at the same time, also the cohort sizes in these plasma studies continuously increase, as proteomics is now not only used for early discovery phase in biomarker discovery and in clinical settings, but also for the later validation stages where the cohort sizes are in the hundreds or thousands of patients. So that leads to a few challenges. So one thing is, so when you want to compare different plasma studies, not all labs around the world have the same instrumentations. And that is sometimes for historic reasons, sometimes for budget, or sometimes just for preference. So the raw data you get out from these different studies will come from different instruments and will therefore be different. And the other is that most laboratories have optimized their own chromatographic systems. So that means they have their specific LC and their specific column materials nowadays, which suits their purpose best, and then they run it there, and this will also lead to differences in the results. Also now separation matrices and column types have increased in recent years, and now more different types are available for specific applications. What this can lead to, you can see here. So this is also when you have like one big study or an overtime study where you want to measure a response, maybe to a diet, that you get one set of samples at a time and then one a few months later. And even if you measure on the same instrument, so your instrument will undergo certain maintenances and calibration, which can lead that your raw intensity of your samples that get out is actually at different levels when you get your data out. And the second scenario is when you have a study so big that cannot even be measured in one site, or it's geographically different, that you just measure on different sites, which completely different instrumentation. The raw data coming out can also be at different levels at the time. One solution to this, as Fabia already presented, is the use of stable isotope standard peptides. So this is a normal scenario where you have only a light channel, and on the left you will see the intensity, and that intensity can actually vary depending on the state of your instrumentation. But when you spike in a heavy isotope standard of a known amount, actually that standard is exposed to the same variation coming from your LC and NS system. And by using the ratio of it, you already normalize within your sample for these high variances in your workflow coming from the LC and MS. And Fabio already introduced PQ500, where we have this set of 800 stable isotope standard peptides covering a good part of the human plasma proteome. Just want to highlight again that these peptides are really coming from an experimental setting where we tried to filter down to get the set together. Intensity was an important criterion, but not the only one. We looked at several different parameters to really get this set of peptides together and to be able to use it directly and easily. So for this study, I will present you now. We used the SureQuant targeted quantitation workflow, which we also called in the past the targeted proteomics on the discovery scale. And I quickly want to introduce this. So this is available since now 2.5 years, only on thermo instruments, on the Orbitrap Fusion series and the Explorers 480 series. It's not really a classic targeted workflow because it relies that you find your heavy peptides, and then when the heavy peptides are found, you only measure your light. So there's all the hassle of it. Targeted workflows like MGRAM and PRAM, they need to schedule your method, and make a new method when you measure is not required. And PQ500 kit is actually pre-programmed on the Fusion instrument and the Explorers 480, so running it is as easy as setting up a DDA method. So it's really one click, plug and play, and you can go. So why is the SureQuant method suitable for our workflow in this case? I quickly walk you through. So you load this one method, it's one click. And how it works is it will check an MS1 level for your heavies. Once it detects the mass of your heavies, which are in a fairly medium abundant range, it will trigger a low resolution fast MS2. And only when it matches the fragments of your heavies, it will trigger a high resolution slow MS2 for your target, for endogenous targets. And this combination is really beneficial that we can have so many scheduled heavy peptides in one run, and also be sensitive with the high resolution MS2 for our targets. And for completeness, for data analysis, I used our own software solution, SpectraDive, which can analyze end-to-end all types of targeted data, including SureQuant, where you load the raw data and then everything gets processed automatically. So the peak picking happens. There's an FDR-based scoring test to correct peaks. The quantification automatically happens. There's some adjustments you need to do for SureQuant, and then you do get your absolute quantification out where you can do your follow-up statistical analysis, which can be also done in SpectraDive. But now to the main part, our small multi-site study. So from study design, we had 7 prostate cancer patients, males above 60, which were confirmed by a PSA ELISA. So PSA is prostate-specific antigen. It's an important biomarker for prostate cancer biology. Actually, every male has it in his blood at low levels, but when you get prostate cancer, it increases a lot. And that is how it's commonly diagnosed. We had 7 healthy men, self-declared healthy as controls. And what was very nice in this, that we could get plasma and serum from the same blood draw. So at the same time, we could also compare how plasma and serum work and if there are any differences. And we also use plasma and serum pools in the study as quality controls. And these, in total, 28 samples, we processed them all together in parallel 96-well format. And then we essentially sent them out to 7 fully independent LC-MS systems, where they were spiked in with PQ500 shortly before acquisition. A few words on these LC-MS systems. So it was, they were all in good QC state. So they were all QC to be working well. It was different MS. In this case, we were limited to Fusion and Explorers instruments. We actually developed the SureQuant method once. So we shared this method. It's a 2-hour nonlinear gradient method, which was identically run on all systems. The LCs, so it was also nanoflow. The LCs were also QC'd, but they were using different columns, sometimes so different column materials. And it was definitely on different days because it took me, I think, more than 4 months to get all the more than 200 runs in that I could analyze them. And then we wanted to answer a few questions. So first of all, how reproducible is this data when we run it on several sites? And for that, we looked at the more easy comparison, the plasma pool versus the serum pool. And when I look at just one instrument, which is here called J, we can see what we expect. So we find certain clotting factors and fibrinogens only in plasma. It's not in serum anymore. And we find it at a certain fold change and Q value, actually. And then when I overlay now the results from all the 7 different systems, you can really see that all these 7 dots at the top left is all this one fibrinogen. The 7 dots below is the other one. So that across all these systems, we really retain not only the fold changes, but with our whole pipeline, also the p-values. So the results you get out are really consistent in this setting when you compare plasma versus serum. Then we wanted to see a bit deeper. So what is really the influence of using these several different systems? And overall, we actually, across all these systems in these samples, we could identify 480 proteins represented by 695 peptides on all of these 7 systems. And when I look into the individual systems, the median CVs are really low for the absolute quantifications. They are between 2.5 on the best instrument, 7.5 on the instrument that had a bit of a worse day, in a single LC in the system. But essentially all the assays, nearly with a few ups and downs, are below 20% CV in technical triplicate injections. And so we had 3 technical triplicates on all instruments, and then not only look at these 3, but have all the 21 from 7 systems. Obviously, the CV increases because I'm looking now at 21 runs, but it still stays quite low at 30% when I look at the pool across all 7 systems. But here you can actually see that there are a few outliers, which are here marked by CV above 100%. And there I went back into the raw data and checked. I can really see in these specific systems have specific interferences, which are easily visible now that you have this whole set of data, but it's just very few. So when you look at between 2 different instruments, it's less than 1% that you really see one interference that's only showing up on one system. Could take it out. I didn't take it out for this. And when you add 7 systems, it adds a bit up, but overall, it's a very minor effect that the individual instruments add. And now coming to the biology, as I already introduced, looking at the prostate-specific antigen, here one specific peptide we monitor, and now you see it across all the different systems. And you can already appreciate that actually they're a bit different scales, but the actual intensity is varying across the instruments in the channel, but it's varying in the heavy and the light. So we can really capture this. And also, especially here in the instrument O, when the LC does not have a good day, you can see it's in the heavy and the light. So consequently, in the absolute quantification, that does not have a big effect. So what you can see here is across the 7 systems, now in one patient, the quantification that the light channels, they are actually varying a lot. And I have to say, in this case, it was independent analysis of the light intensity. So in a discovery setting, you would normalize and then get some of this variation out. But here it's just the raw light intensity. And actually nicely, the heavy peptide synthesis is quite abundant and the target is very low abundant, but still we capture both. And in the heavy to light, in the absolute setting, this is really taken out and we can nicely quantify this fairly low abundant biomarker, the CV below 20%. The question is now, does all this variation between the instruments, so is it now relevant which instrument I use for the biological results I get out? So when I use the wrong instrument, would I get a wrong conclusion? And for this, I looked at in this case, the best performing instrument was instrument J in Expose 480, where we can actually nicely detect 2 peptides for prostate-specific antigen. Here you can see also plasma and serum next to each other. And this is not an outlier for most proteins. We really see the same profile in plasma and serum. You can overlay it, except for the ones that are not in serum anymore. Fallen out at higher levels, so significantly different in cancer, lower levels in the healthy. And I would say the worst instrument in the study was the fusion Lumos, where we have a lot less, or we have quite less sensitivity. So we don't really detect it in the healthy, and we only detect 1 peptide in the cancer patient, but we still get it significantly regulated on all systems. So it's really, when we find it, the quantification is really accurate and we get a good result. But the sensitivity is, of course, different between different systems. And we were motivated by these results, but we wanted to get still a bit deeper on the healthy patients. So I ran one additional study where we depleted the whole sample set and only ran it on 1 instrument and analyze results. And when we deplete it, we actually find it nicely in all healthy patients as well. We can measure PSA levels. And as I told you in the beginning, we do have ELISA data from the cancer patients. And when I now look at the absolute amount of PSA we measure with SureQuant in this data, And the ELISA data we have with it, it's actually very, very consistent. And this is nanogram per milliliter plasma. So this variation is not basically nothing. And I don't really know which assay here, but where the ground truth really is. We don't have data for the healthy patients, but what we can say, if you look at the CDC guidelines, healthy men above 60 should have below 4 nanogram per milliliter, and we actually measure below 4 nanogram per milliliter. So even when we further validate against non-MS assays, the results make really sense. And this brings me to the conclusions already, that one is, so the MS acquisition with heavy peptides really directly creates a digital record of the biological results of the plasma and serum samples. So there's sometimes read that you need to validate mass spec data against some other published or ground truth data. This is, I think, really not necessary, because mass spec really directly gives you ground truth data out of your biological samples. The instrumentation itself only marginally influences the quantification results. So you've seen this is less than 1% instrument individual effects, which do not really have an effect on the statistical outcome on an experiment. What the instrumentation does affect is the sensitivity analysis. So when you use different instruments, you might see more or less, but the things you see are really reliable. That's the main message. And I think this whole workflow will really be an important tool for population scale plasma or serum studies that we now see coming more and more in the field. And then they can directly compare results across labs and sites. I finish. As Fabia told, there will be a Q&A after the whole seminar, and I'm happy to hand over to Manuel for the next.
Thank you, Sebastian. So I have the pleasure to talk to you about our work on COVID-19, which started almost exactly 2 years ago, so during the first wave of the pandemic in the UK. And I will show you how we compared protein biomarkers against RNA biomarkers, in this case, RNemia. So the title of the talk is SARS-CoV-2 RNemia and proteomic trajectories inform prognostication in COVID-19 patients admitted to intensive care. Our main focus were patients that had COVID-19 and were submitted to ICU units. We collected from 78 patients, almost 300 samples at baseline, week 1 and week 2. And in addition, we had, of course, controls. These were non-ICU COVID patients, but also ICU patients that were also in intensive care, but for other reasons than COVID-19. And I think this was an important discriminant in our study, that we actually also had sepsis patients as controls. And we didn't just compare severity, but we compared basically our protein and RNA biomarkers to mortality in these patients. So the first biomarker is an RNA biomarker. It's the detection of the viral RNA in blood. We do this using a 2-stage qPCR and RNA extraction. We then compared the RNA biomarkers to proteomic biomarkers, which were validated by ELISA. And for this, we're using the PQ500 because we think it's essential to have authentic reference peptides in these large cohort studies. And for data analysis, we use Spectronaut. In addition, we did some functional analysis where we used the SARS-CoV-2 spike proteins in pull-downs to identify plasma proteins that bind to this spike protein. So in the first part, I will talk about detectable viral RNA in blood. And the summary is here, only 4% of non-ICU COVID patients have detectable RNemia in blood. So you have to become very sick for the virus RNA to be detectable in the circulation. But if you go to ICU patients, approximately a quarter of them has detectable RNemia, and this is associated with really poor outcome. So if you are RNemia positive, half of those patients die compared to 13% who are non-positive for SARS-CoV-2 RNemia. So RNemia is especially more frequently detected at a time when your antibody response is still low. So we detect it within the first days of admission to ICU, but once the antibody response kicks in, RNemia becomes less detectable. So in terms of biomarkers, we all know that age is one of the best predictors for mortality in COVID-19. However, on the other hand, many other clinical variables we are using for risk scores to assess mortality in sepsis patients don't work very well in COVID-19 patients. And this is what you see here. So on the x-axis, you've got the hazard ratio for mortality. And on the y-axis, you've got these different clinical risk factors we usually use in risk scores. And most of them, apart from age, are not associated with outcome in COVID-19. But you can see that anemia is actually almost equivalent to age in terms of predicting mortality. Even if you correct for age and sex, anemia remains statistically significantly associated with outcomes. Anemia, of course, has one advantage. It has got 100% specificity for the disease. So we won't find a biomarker at the protein level that's better than anemia. However, there's a problem, as I mentioned, that anemia is only detectable at the early stages of the disease. And once the antibody response kicks in, anemia becomes undetectable. So we need additional protein biomarkers to monitor those patients over the time course of the disease. And for this, we used DA-based proteomics. As I mentioned before, we were using the PQ500 kit, and we treated our samples with the WHO recommended detergents at the time. So that was 1% tritone. We didn't deplete the top 14 proteins, and we had like a 1-hour gradient to basically monitor as many proteins as possible, and then analyze the data by spectronaut. So our study wasn't the first study in proteomics on COVID-19, but at the time, most of the previous proteomic analysis always concentrated on COVID severity. So they always compared healthy individuals against patients with different disease severity. And there are 2 publications highlighted here, but I think the key comparator was always healthy controls. Now, our aim was to have, first of all, a longitudinal assessment, so get multiple samples from the same patients over time, and then compare the proteomic changes, not to severity, but to 28-day mortality. So the way we prepared the samples is using an automatic robotic system. This is a Bravo robot from Agilent. We used 10 microliters of plasma. We also had to do an automated C18 cleanup because, of course, we had a lot of detergent in those samples. We spiked the PQ500 heavy reference peptides, which also have the IRT retention standards, which are really essential if you do these large studies. And then basically run it on a Fusion Lumos, actually. So maybe we should have run it on an Exploris, but I'll talk to Sebastian afterwards. And then for data analysis, we used Spectronaut, which is really very user-friendly. And the first take-home message is that protein biomarkers that are associated with severity are not necessarily good predictors for mortality if the patient is already very sick. So this is a list of proteins that have come from those 2 previous publications that link these proteins with disease severity. But if you look at these patients who are admitted to intensive care, who are all severely sick, then those proteins actually don't inform on their mortality anymore. And the reason, basically here, some of those proteins, complement factor B and carboxypeptidase N and alpha-1 antichymotrypsin, so are 3 proteins which were validated, but many of them were not. And the reason here is shown with these sepsis patients as additional control groups. So here you see a heat map for proteins that are upregulated in COVID patients compared to control patients. And of course, if you compare very sick patients against healthy controls, we always find a lot of differences in proteomics. The problem is they lack specificity. If you actually have another comparator with equally sick patients that have not COVID-19, then you find fewer differences, but these differences hopefully are more specific to the disease. So I would argue that in many proteomic studies, I think this is a very common mistake to make. We always have healthy controls as a comparator, and we are very happy that we find many differences. But as a clinician, I can tell you that those differences are markers of sickness, and I wouldn't use them in the clinic because they're not informative for my patients. So what were protein biomarkers that are interesting from a pathological insight? I think 2 proteins that I would like to highlight. One of them is called mannose binding lectin 2, and it's a very important protein in the lectin pathway of complement activation. Complement, by the way, was one of the enriched plasma protein clusters that was associated with mortality. And mannose binding lectin-2 was associated with a higher risk of mortality. It binds to the sugars of surfaces of bacteria, fungi, and viruses. And of course, this is highly relevant for the coronavirus because the name corona refers to the distinctive crown of glycoproteins on the viral surface. Now, what does mannose binding lectin do? It actually interacts with two proteases. These proteases are called MASP1 and MASP2, and MASP stands for MBL-associated serine proteases. So to give you an overview about the complement system, you're all familiar that we've got the classical complement cascade, which is induced by antibodies. But then we have this alternative complement activation with the lectin pathway. And MBL2 sits right in the middle here. It forms this complex with MASP1, and that modulates the alternative pathway of complement activation. But importantly, it also interacts with the coagulation cascade. And we know that our patients with COVID-19 have thrombotic complications. So once you get activation of the MBL-MASP complex, you get also basically cleavage, for example, of prothrombin to thrombin via MASP2, and cleavage of fibrinogen to fibrin via MASP1. So the complement activation via this pathway is also activating coagulation. The other protein biomarker that predicted 28-day mortality was pentraxin-3. And again, this has been externally validated in many other cohorts. By now, it's a marker of neutrophil degranulation. And interestingly, pentraxin-3 is actually part of the innate immune system, which links the lectin pathway and the classical pathway. Again, because we encounter a new virus, we didn't have antibodies. What pentraxin-3 allows you to do is activate the C1q complex in the absence of antibodies. So it's an innate immune activation system to activate the complement cascade. So MBL binds to pentraxin-3, and the formation of this MBL pentraxin-3 complex recruits C1q. So we found these two proteins particularly interesting among the proteins that were identified because we think they are functionally related. Also based on our previous data in non-COVID patients. So when we did a study published at the time in molecular cellular proteomics in sepsis patients, and in fact, this was data from a mouse model of sepsis, we could see that one of the proteins that accumulated in the tissue was pentraxin-3. It's stained here in green. This is a cardiac tissue, and you can see these small capillaries. And what you're seeing here is how the neutrophils, when they extravasate, they degranulate, and they deposit pentraxin in the tissue. So you don't just have inflammation in your blood. The cells in the blood carry, of course, these proteins into your tissue. And actually, that was shown in a subsequent study where we looked at LPS-induced endotoxemia, both in men and in mice. So we had healthy human volunteers who were given a very low dose of endotoxemia. We had serial blood samples, and we could show that pentraxin-3 elevation in response to LPS is linked to the upregulation of many other neutrophil-derived proteins in the blood. But then, of course, in the mouse model, we could also take the tissue, and we could show that if we take the vasculature, in this case, the aorta, that pentraxin-3 is one of the few proteins that's not just elevated in the blood, but it is also elevated in the tissue and therefore can mediate inflammation-induced organ damage. So if we look at Kaplan-Meier survival plots for 28-day mortality for anemia and pentraxin-3, then as I told you, for anemia positive, half of those patients unfortunately die. If we look in comparison for pentraxin-3, the mortality curve is comparable, but maybe slightly less compared to anemia. But importantly, what is, I think, really relevant is that both of these biomarkers have since extensively been validated by other investigators as being predictors for outcome, both anemia as well as pentraxin. For pentraxin, I need to say how important it is to consider serum and plasma, because pentraxin levels are different, whether you use serum or plasma as a matrix, because of course with serum you coagulate the blood, and when you coagulate the blood you also basically get an activation of neutrophils which release pentraxin. So your serum levels and your plasma levels are not directly comparable in those two matrices. But anyway, we then used a machine learning approach where we fed in all the clinical variables and all the measurements we had. And again, this was just the outcome of this machine learning model where the best binary combinations were age plus arenemia and age and pentraxin. So the final part was functional analysis, what plasma proteins can interact with the spike protein. For this, we used affinity proteomics, the spike protein for plasma pull downs and analysis by mass spectrometry. So as you know, the spike protein is the key protein that allows the virus to attach to the host cells and enter the host cells. And the protein that we pulled out from plasma is one of the spike protein binders, is galectin-3 binding protein. It is highly expressed in the lung, and it is known to possess antiviral activity. Again, why is that interesting? If you look at the SARS-CoV-2 spike protein and its N-terminus, and you compare it to galectin-3, you can see that there's a high structural similarity. And the concept is that the virus uses a dual attachment model to enter the host cells. The key interaction, of course, is the interaction of the C-terminus of the spike protein with the ACE2 receptor. But the N-terminus, because of its structural similarity to galectin-3, may actually engage certain sugar binding receptors on the cell surface. So the question is to what extent this interaction is functional for the uptake into the host cell. So first of all, we try to validate as many of our proteomic changes by independent measurements, in this case by ELISAs. So this is just the result for galectin-3 binding protein, where we again compared COVID-19 ICU patients against sepsis ICU patients. This is the mass spectrometry-based quantification, and this is the ELISA-based quantification. And the point I would like to make here is that for me, the difference that is important is not the one between the controls and the COVID patient, but the difference between the sepsis ICU patients and the COVID ICU patients. And what it indicates is probably that galectin-3 binding protein is predominantly induced by viral infections, whereas our sepsis ICU patients, many of them had bacterial peritonitis, for example, or bacterial lung disease. So finally, the key question is, does galectin-3 binding protein have antiviral activity, at least in vitro? For this, we use two assays for functional validation. One is a spike-induced cell-cell fusion assay. The other one is an assay where we use spike pseudoparticles for entry into the host cells. The cells we are using are HEC293 cells, and these are the results for the spike-induced cell-cell fusion assay. So what you are seeing here in green is cells that are expressing the spike protein. And once the cell is expressing the spike protein, they're actually fusing with the neighboring cells. So this is called syncytia. We also see that in patient material. It's a characteristic feature of COVID-19 that you get these fusions of cells, this syncytia formation. And if you overexpress galectin-3 binding protein, here highlighted in purple, then we can attenuate these fusion processes. And as a positive control here, we use as RNA against ACE2. If you knock down ACE2, again, this fusion is blocked. The second assay is a spike pseudoparticle entry assay. So we have got the spike protein on the surface of the particle. If the cell is taking up those pseudoparticles, they are highlighted in green. And what we are seeing here is overexpression of galectin-3 binding protein in red. Again, reduces the uptake of these spike pseudoparticles. The most efficient way, of course, is knocking down the ACE2 receptor. And as additional controls, we had here proteins from another virus to actually show that this mechanism is specific for the spike protein and doesn't occur with other virus entry mechanisms. So this is the quantification. So for functional validation, and this is of course all in vitro, we had two assays, the spike-induced cell-cell fusion assay with galectin-3 binding protein being protective against cell-cell fusion induced by spike. And similar for the spike pseudoparticle entry assay, overexpression of galectin-3 binding protein attenuated the uptake of these spike pseudoparticles. And ACE2 here as a control, as a positive control, and the non-targeting as RNA as a negative control. So with this, I'd like to conclude. The take-home messages is that arenemia within the first days of admission to ICU is an excellent biomarker for 28-day mortality in those patients. Its strength is comparable to age, and because it's the viral RNA, it has 100% disease specificity. On the other hand, of course, proteomics was very powerful because it was an unbiased approach to identify novel protein biomarkers. But as in many of our studies, the control group, I think, is the most important decision. And I would argue that we should have other diseases, comparable diseases as a control group, and not healthy controls, to get more clinically meaningful biomarkers. And in our case, we used other sepsis patients as a control group. And some of them also had acute respiratory distress syndrome. Then we used the machine learning approach to integrate the information from the RNA data, the clinical data, and the proteomics. And the best performing clinical characteristic was, of course, age. But we could show that actually adding an RNA biomarker or a protein biomarker on top of age significantly improved prediction for 28-day mortality. What we like about mass spectrometry, I think that's why we all at US HUPO, is of course that mass spectrometry-based proteomics is unbiased and it is applicable to a wide range of experiments, not just to plasma proteins, but to pull downs and so on. And in these pull downs, we identified this galectin-3 binding protein as a potential novel binding partner of SARS-CoV-2 spike protein. And finally, I think we need companies like Biognosys to actually help us with our research. We couldn't make all these reference peptides by ourselves. But for large clinical cohort, I think it's essential to have these controls, especially if your samples are spiked with nasty detergents and you have a lot of runs and samples to get through. So for us, it was essential to actually have these heavy reference peptides to get reliable quantification and then have external validation by ELISAs in addition. So these are my acknowledgements. I have to really thank a lot of people. This was a real team effort because everybody was coming together at the time because all non-essential research was halted at King's College during the first pandemic, which meant we all redeployed to COVID research. And it was a really very fruitful collaboration. And I would like to thank all the people, especially the intensive care physicians, the team of Dr. Manu Shankahari, who not only looked after the patients, but in addition also provided us with the samples. Thank you very much for your attention.
Actually, I will invite both our speakers here and open the Q&A session. Please, Birgit.
Yeah, this was really wonderful, both of you. Thank you very much. I had a question maybe to Sebastian. Or maybe two actually. So I assume it's a scheduled method, right? With the SureQuant?
No. SureQuant, you don't need any scheduling. So you just, when you detect the heavy, the light gets measured. Yes. So that's why it's so easy to transfer across chromatographies in this case.
But now that I asked, well, we've done it ourselves, so it's not scheduled. But so then how do you see, I mean, potentially one could expand upon the PQ500, right? And add extra reagents and all this, right?
Yeah. So PQ500 with the 800 peptides is already, so we did the 2-hour gradient in the end. So SureQuant, because of the setting that you first need to detect your heavy and then your light, you need a certain measurement time. Like it's not a super fast, short gradient, super high throughput method. Right. But when you add more, of course, you then should also extend a bit your measurement time, but that should work.
Maybe then I ask my second question. So when you compare the SureQuant versus if you did it in DIA, how much difference do you see? A lot of difference in sensitivity, or is the DIA?
We do see some differences. I think I had this. So DIA, because you also measure all the other peptides in the sample, you definitely you lose a bit. You end up a bit more shallow or pure. In plasma, it's always hard to really quantify this because you would need like a reference set where you can then really see where are you in. But I would actually say that it's really hard. So maybe about 10x further with SureQuant in, but then you don't measure all the other. And with DIA, you have the advantage you measure all the light in your sample until your limit of sensitivity, but you have this additional information which you get out of it.
So would this be a limit of detection or quantification?
So, I mean, this depends on each specific assay then, and also, like as I said, we measure this once, or we measured it for MRAM, for example, and SureQuant is a bit more difficult to measure it. We do see it go down in the nanogram per milliliter range for plasma. So when you for the original plasma concentration, that's what we see. It always, yeah, it's plus minus also tenfold for each individual assay. But we do have data, and for MRM we have a list on our website. You can check.
How much is that dependent on how much you actually spike in into the plasma?
No, the spike in is always the same. It is a bit dependent on how much plasma you use. So that depends again on your mass spec, how much is in ideal range if you use a bit more. But also mostly on your analytical setting, like how much time do you spend on your sample, how long your gradient is. There you can push it sensitivity a bit more. If you want to have high throughput, you will get you have less sensitivity, but obviously measure way more samples.
And how much is the identification of these peptides dependent on spectrum length?
So for PQ500 itself, we provide a ready-made library. So there's actually no dependence. So you don't need to make your own library. So you get it for these. Yeah, so you can combine it with a label-free library in Spectronaut. You just use both in the end, your label-free library and your PQ500 library. There, if you make a label-free library, it's definitely recommended to make it from samples not spiked with PQ500, that you don't get interference from the heavy peptides, but otherwise it should work. And Spectronaut is made to figure out which entry to use from both libraries. Yeah.
How many samples can you process with one kit? And did you see any variation between kit to kits?
So we have several different versions between 24 and 96 samples per kit, and we did check this over time, and we didn't see. So it's all coming from one batch, the heavy peptides, and so it's all fine. If you like, yeah, the vials are kept frozen and dry, then it's all good.
When you compare like the liquid sample versus non-liquid samples, any of these peptides do you usually identify, like the corresponding right peptides do you identify in a heavy sample?
So when you compare native versus depleted, how much more you get? So actually with SureQuant already in native, we get a lot, as told this 480, and with the depletion, we do not really add so much more because we're already at that's what the kit is. We get slightly above 500. But what we really do see that what you maybe also noticed in the CV plot, that on the lower edge, we do have higher CVs because we are at the limit of detection, and these get more stable when we deplete. So the number of IDs does not get so much more, but the variation of the lower abundant peptides gets lower when you deplete.
800 peptides are from 800 different.
They are from 580 proteins, so we have some proteins with several peptides. Yeah, so they are all, yeah, all over. And some of them you really need specific, like they're for specific conditions, like heart conditions or PSA, you will only find in prostate cancer patient, the male. So it's not expected that you find all of them in all phenotypes. So some you need specific ones, but I think with depleted, you basically get all of them in your sample. Yeah.
Can you talk about the performance of these kits in both like targeted or DIA setting in non-Orbitrap instruments?
Yeah, so actually they also work quite well. So especially on these latest TOF instruments, so XenoTOF or timsTOF, they do work well. You especially on short gradients. So there you can really get a high throughput and short gradients. At what data I've seen, you do not get to that level of sensitivity, but still to a good level of sensitivity, but in a fraction of the time. Like also in MRM, you will end up you can do half-hour, 40-minute runs. You will not get 480, but you get 300 proteins still identified in your light, but you have a way higher throughput. So that's always the two things you can dial. You can decide how much time you want to spend. If you want to have high throughput, then this TOF or MRM instruments is your choice. If you want to go really deep, so far this workflow on the Orbitrap instruments is the most deep we saw or I saw.
MRM does outperform DIA is also.
MRM and DIA is actually quite at level, but at DIA you need to be a bit longer gradients to achieve the same. Yeah.
What was the throughput of the studies that you?
This was 2-hour gradient, so sample to sample 2.5. So yeah, 10 samples a day. So took 3 days to acquire a set of 28 samples. So not super high throughput.
But I found very powerful, of course, that you did this at multiple different sites, right? And
so multiple different instruments. Yeah.
in the same lab?
Not in the same rooms, but yeah.
Right.
So we only worked in our thinking. Yeah.
But supposedly one could do it in different
Yeah.
sites,
So
right?
actually wanted to model it as much as possible on different sites. So that's why it was over a course of months.
Yeah.
The instruments were maintained at different they all passed basic QC parameters, which we defined before, but otherwise it was really different operators and so on. Yeah.
Because there are some developments by, say, CPTech, right, where they really encourage having it in different sites. And you mentioned also different cohorts potentially, right? So I think, it's one kind of strength potentially in the mass spectrometry field, right? That people can collaborate like that and really validate things very independently, right?
Yeah, it's fully agree.
I have a question for you. So in one of your slides, you compared the, I think it was categories in measurement using ELISA as well as mass spec. For ELISA, how do you normalize the data?
Well, for ELISA, I mean, the advantage, of course, of the ELISA is that it has a protein standard, right? So I think that's one of the problems we are still lacking in the mass spectrometry field. We have peptide standards to quantify proteins. And I think what's nice about the Biognosys kit is that they've pre-selected the peptides, which are reliably detected. But what, of course, an ELISA provides you is an absolute quantitation based on a dilution curve of a protein. And that's why we often still kind of cross-validate our mass spec data with the ELISA data, like you have also seen in Sebastian's talk. Because I think peptide-based protein quantitation compared to protein-based protein quantitation may still sometimes have some differences. Yeah.
Sorry. Oh, sorry. So just one more question. For your expressions level of MBL, how much was that correlated with different risk factors, let's say diabetes or immunosuppressant individuals?
Well, actually, MBL is a liver-derived protein. And what you see in sick patients in general is a suppression of liver-derived proteins in plasma serum. So what you notice in many of the studies publishing on COVID severity, for example, is that you get a reduction of many biomarkers. And that's because the liver is our main factory for producing plasma proteins. Many of them are detectable within the mass spectrometer. If you're very sick, the liver doesn't invest that energy in producing more plasma proteins, and therefore you get the reduction of many proteins. That's why for us, MBL2 was particularly interesting, because the liver-derived protein, despite the suppression of liver proteins, we're actually going the opposite direction. So the liver must have made that decision to basically produce that protein at the expense of other proteins. So I think the major factor was not comorbidities, what we saw. The major factor is that in patients who are at ICU, you get a suppression of liver function and therefore reduction of many liver-derived proteins. You can actually nicely show that in a pathway analysis, that most of the proteins you see being reduced are the hepatic function. It actually is linked to hepatic function. So that's what we are seeing in the plasma proteome. So again, I think that's where the challenge is in proteomics. And I think also the exciting aspect of it is like a bit of a detective work to figure out what actually is going on. And you have a lot of background noise, a lot of protein change, but some of them may be more informative than others. So yeah, I think the reduction of liver function was the main parameter. You can also see that in liver transaminases. So if the patients are very sick, the liver transaminases go down. And on the way to recovery, when they're actually getting better, then the liver proteins rise again. And the same is actually true for liver microRNAs. So it is just a readout of your disease state.
So actually, I have two questions for you. My first question is, did you stratify your COVID-19 patients for your analysis? And my second question is, did you actually follow up this patient for possible long COVID symptoms?
Yeah, so I mean, these were, we didn't stratify them. We basically had all comers to the ICU, pretty much all patients that were admitted to ICU at King's College in this first wave. We got samples from longitudinally, but basically it was an unselected all-comer COVID patient cohort. But we had the hospitalized COVID patients that were not in ICU at the same time, and sepsis patients from the pre-COVID era. So these samples were taken half a year earlier, so we could be sure they didn't have COVID when they were admitted to ICU. And we didn't follow them up for long COVID, but as you know, there are several large cohorts now going on to look at what is long COVID. It's still a very puzzling picture, which we are seeing. There is no biomarker or no diagnostic way actually of establishing whether somebody has long COVID. But yeah, it is because of the heterogeneity of the disease. It's also, I think, a very tricky one to tackle. I mean, the patients report very different symptoms. So I think there's a lot of work to be done, but we didn't follow up those patients on long COVID. We need to go to different cohorts.
All right. You want to take the last question?
I think it's always good.
Yeah.
One quick question.
Last question.
Did you gather any time series data, and how do the levels of PTX3 and MBO2 change over time?
Yeah, short answer is yes, we did. So we had these longitudinal samples, and that's why I said like we could make these trajectories for proteins, how they changed during a recovery phase in patients who survived, and how they deteriorated or changed in patients who died. It's all in the paper, but having these longitudinal measurements is important because we don't just want snapshots. We want to know how our biomarkers performs in patients over time.
A round of applause to our speakers again, and thank you very much.